Akansha, how are you? Thank you very much for all your help.
Yeah, Dr. Iqbal, thank you very much for joining.
Yes, sure. Anytime.
I want to introduce Rajeshwari Jayakumar. She is at VCU Health, Virginia Commonwealth University, and she is practicing hematopathology right now. She is gonna be one of the speakers with me. Like, we are going to discuss together, and of course, this is more going to be a discussion-based sessions. This is how our HEMPAT WHO book clubs were. They were more of discussion-based sessions, and we are going to learn together. Basically.
Okay, good. Thank you very much.
Yeah, and whenever you have
introduction, Akansha.
Yeah, please go ahead.
Thank you.
Someone was saying something?
No, I said thank you for the introduction.
Of course, yeah. Rajeshri, you want to share your screen? Let me let you to make co-host.
Yes. I'm just opening my PPT. Okay. Okay, is everyone able to see my screen?
Yes.
Okay. All right, so I thought we'll first start by going through what happens in the lab before we see the different colors in our screens, because many people have some knowledge gaps on how the flow panel is being set up. So just wanted to address that before we go into the discussion on how we analyze things. So there are many samples that we get, ranging from peripheral blood to bone marrow, body fluids, lymph nodes. But the most common sample that we all get is peripheral blood and bone marrow, and the accepted anticoagulants are mostly heparin and EDTA. Although, like, the literature says about ADA and other anticoagulants, but I don't think anybody is actually using it in their laboratories in common practice. EDTA and heparin are the acceptable specimens. And once we get, let's assume this is a bone marrow specimen, and I'll just walk you through the procedure of how it is done. Let's assume we get a bone marrow specimen in a heparin tube, and then the lab would isolate some of the specimen into this falcon tube, and then they will then first do a wash step. They'll use bovine serum albumin, which is in colloquial language referred to as stain buffer. They'll wash it in a couple of times, and then some laboratories, they first lyse the red blood cells if it is a bone marrow sample and then wash it. In our laboratory, we first wash it. Then they centrifuge it, and then they make a pellet. And also simultaneously, they make an antibody cocktail. So just to let you know what an antibody cocktail is. So for each analysis, let's say, for example, bone marrow analysis, we would need a set of tubes for it. So depending on how many colors your instrument is capable of analyzing at a time, we have ranging from 4 to nowadays we have even 16 or more colors that we can analyze at a time. Ours is an 8-color laboratory, so we use 8 antibodies or fluorochromes at a time. Antibodies are what we use for analysis, like what we need for analysis, like your CD45 or CD38 or CD34 or all those clusters of differentiation that are targeted against the antigens expressed on the surface or in the cytoplasm. Those are your antibodies. But that is not enough for fluorescence. So that is tagged with the fluorochrome. And the things that you see labeled here on the top are the fluorochromes like FITC, PE, PERCP. All these things are fluorochromes. So the property of the fluorochrome is to be excited by a laser light of a particular wavelength at a particular wavelength. And then when they come back to their normal state, they emit fluorescence. So that is detected by the detector in the flow cytometer, and that changes that into a pulse, and then you then get the events that are either positive or negative for that particular fluorochrome excitation. So any questions with the fluorochrome or what happens with the antibody so far? All right. So then we make antibody cocktails. So we have different antibodies tagged to fluorochromes separately, like for example, CD45 with APC or CD45 with FITC. And so we have each fluorochrome tagged to one antibody in different combinations. So then you select 8 different antibodies. Like for example, in one of our myeloid tubes, we have CD38, CD34, CD117, CD13, CD33, and CD11B is CD15. So we have different combinations in different laboratories, whatever you want to see. And then you put all those, like for example, if your instrument is 8 color, up to 8 color, you can add in one tube. So it may not be enough to analyze all the markers that we are interested in analyzing. So we may set up more than one tube for myeloid. So like 2 tubes for myeloid, 1 tube for B cells, 1 tube for T cells, or some people just do a mini panel. If they think this is a normal marrow, they just set up 1 tube based on what your requirement is. Basically, the bottom line is you can put as many antibodies that your instrument can analyze in instrument has the capacity in 1 tube. If it is 8 color instrument, you can put 8 antibodies in 1 tube. That's the principle. So then you mix the specimen with your antibody cocktail, and it has to be incubated in the dark because it's a fluorescent antibody. So you don't want them to be excited haphazardly in your room light. So you want them to be stable, and until they hit the laser in the instrument, you want them to be preserved. So you put them in the dark. So what most laboratories do is they just put it in the drawer beneath the desk and just shut it up for 15 minutes. Most likely the incubation is 15 minutes, but based on whatever the manufacturer recommends. Then once the incubation step is done, for bone marrows, we don't want the red cells. So we need to lyse, red cell precursors. So we need to lyse them and take it out. So for that, you use the lysing solution. And so some people tend to skip it in peripheral blood, but even in the peripheral blood, you have mature red cells, so we do it. So then you lyse it. Some people do the lysing step before adding the antibody cocktail. In our lab, we do it after that. And then you incubate it again to let the lyse agent act. Then once it is all lysed, you wash it twice to remove all the debris with the bovine serum albumin. And then now your specimen is ready to go into the machine. But before you start preparing the specimen, it's important you start the machine, run your quality control. So there are beads that are available in the market that people use for QC. So they take these beads, these are like small particles, almost invisible, and they can actually get tagged with the fluorochromes and the antibodies. So you can use your antibody cocktail with the beads. The beads will pretend to be the cells. And then you can adjust the equipment compensation and voltage based on of the photomultiplier diode based on the emission, and to adjust. That's how everyday morning QC is run. Once that is done, you're ready. Your instrument is ready to go for the regular runs. So now your specimen is ready, you're feeding it into the machine, and what happens inside the equipment is this is the laser light. You have different sets of lasers with each emitting a different nanometer light. So and one interesting thing to remember is when you design your panel, a lot of things go into that. We use the brighter fluorochromes with the antigens that are expressed less on the surface. and we use the dimmer fluorochrome with the antigens that are expressed more, just because you don't want if for example, if we are using a brighter fluorochrome to an antigen that is expressed more, it will spill all over and you will see a lot of false positive. So to avoid that, the combination is usually less number of antigens, brighter fluorochrome, more number of antigens, less dimmer fluorochrome. And also we don't want fluorochromes that can cross over nearby. They space it. I mean, the lasers that can cross over nearby, they space it very well in the instrument. So what happens is there is a sheath fluid circulating here, and the specimen that is fed will pass through this tiny chamber where they are in streamlined to make sure that only one cell passes the laser light at a time. So here is the slit where the cells will hit the laser, and when that happens.
No, I don't do that shit. If they're not going to believe me on my word.
Okay. Can you
Sorry,
please
I
mute
muted.
yourself? Yeah, okay.
Yeah, I did.
Okay, thank you. So when the cells hit the laser, they get excited, and whatever happens there is being captured by the detector here, and then that is converted into pulses, and that's what you see in your output. Now when you get in the whatever you get in the output is stored in the equipment, and then that raw data can be analyzed using a software. So there are different types of software like Woodlist, Fax, or Kaluza. Whatever you want, you will import that raw data into your software and you'll analyze it. So labs usually make templates for that. So for example, I'm going to just show you a template. Let me see. Oh, are you still seeing my screen?
Yeah.
Okay, so this is a template, like they will have a fixed, for example, this is a myeloid blast page 1. If you see, if you can see closer, this is for, do you see that yellow box that is popping up? Do you see this yellow box popping up below my cursor?
No.
Okay, hold on. Okay, it just comes for a second and disappears. Now? Yeah, so that shows CD45 versus site scatter. So we have fixed templates in there where you will import your raw data. Raw data is only gray color or black color. You will have to color it based on your gates to to highlight the population you want. So one funny question that I often encounter from residents is like, they confuse the colors that we see in the plots to what colors the equipment is capable of. So let's say, for example, I say 8-color instrument and they see like 10 or 12 colors in the output PDF flow cytometry plot. They think, oh, you said it is 8-color, but I see more than 8 colors here. So the colors that we use for gating is what we give it. We give it after the raw data is out of the machine. So do not confuse that with the colors that we are actually talking about in the instrument. What we are indirectly implicating is the fluorochromes, how many fluorochromes we can use in one run. That's what it means. So now Akansha will show you the gating, how it is done. But before that, I just wanted to briefly talk about the viability testing. So this is something that we do in tissue specimens more commonly, or if the specimen came on a Friday and you're analyzing it on a Monday. So what they do is they use a dye, which is 7-AAD. 7-AAD also is a dye, but it also has a fluorescent property. So this binds to the GC part of a DNA. So when a cell is intact, it is impermeable. So when a cell is intact, the 7-AAD cannot bind to the DNA, right? But when a cell when there are necrotic debris, or if the nuclear elements are out of the cells. If the cells are disintegrating, then 7-AAD can go and bind to that cell. So what they do is the cells that are binding to 7-AAD will be here, that are in the positive area, and they only get and choose the cells that are negative for 7-AAD, and that is what is considered as viable. It is important to eliminate these dead cells because they can stain with all kinds of antibodies and they can produce very non-specific reactions and a ton of false positivity. So we make sure we do it for specimens that we expect to be contaminated or expect a lot of degeneration. Okay, so now Akanksha will show you more. Any questions so far?
Okay, guys. I am Akanksha. So I think that was pretty nice overview of how a flow cytometry is done. Any questions that you have from Rajeshwari that you want to clarify? Okay. So there is a couple of people who are trying to get into Zoom. I don't know what they are doing wrong, but they are not able to join, Alia and Shweta. And I don't know if someone in the background can help them. That will be very helpful. Like maybe call them and tell them what to do. There are some questions in the chat, Rajeshri, if you want to answer.
Yeah, I'm responding to Junaida now.
Yeah, you can answer like just like.
Okay,
by
so
phone,
her
yeah.
question is if we add 7-AAD to all tubes, no, that's a separate tube. We just add 7-AAD and CD45 in that tube, and that is done before you set up anything else. Yes, that is correct. It is in a viability tube, Halima. So we run that, and if we do not see enough viable cells, we don't do anything else. We just say inadequate specimen and stop there. It is done in a separate tube.
All right, cool. So I want to quickly show where everything lies. So this dim CD45, dim site scatter. So first thing first, what is site scatter? So, I mean, I know that there are people who are starting from scratch, and there are people who are very advanced. So, but we'll be starting from basics, and then we'll go to more advanced. So basically, what we are going to do is we are going to have these introduction sessions, 3 introduction sessions. 2 of them will be tomorrow morning for us, and 1 of them is today. And basically, we'll be going through basics first, like the basic that Rajeshwari showed us, and also the basics of gating and stuff like that. And then we'll be diving into the real cases. So the fun will start eventually. So site scatter, what is site scatter? Site scatter is the complexity or granularity of a particular cell. So if you, and what is a forward scatter? Forward scatter is the size of a particular cell. So for example, the site scatter of granulocytes, as we know, there are more complex, they are more granular. The site scatter of granulocytes is bigger, right? So that's what I was coming to, that this population is blasts. Why? Because the site scatter is kind of low, and in blast we know that there are not really much of granules, and also CD45 is lower than this population, which is lymphocytes. So this is B and T lymphocytes, and you can see the CD45 is kind of bright in the lymphocytes, and it's kind of dim. It's kind of dim in the blast. So that's the blast gate where it lies. And as I mentioned, the neutrophils, they have very high site scatter, but again, the CD45 is like dimmer than the lymphocytes. The brightest CD45 is for the lymphocytes. Then coming to the monocytes, so monocytes have like less granularity or less site scatter than the granulocytes, and they lie in between the blasts, grans, and lymphocytes, if that's what you want to remember. Then these are the B cells, and again, as Rajeshwari is told, that the debris we have to remove out, and this is the RBCs and debris where it lies. So this is a nice WHO chart that is given in our WHO book, and though it looks very busy, but this is a this is a very, very important chart that we have to remember whenever we are doing flow cytometry. Let's review the common things first. Maybe I can show, no, actually not. So maybe, so let's see. So again, this is busy, but let me orient you. So hematopoietic stem cell, common myeloid progenitor, granulocyte, monocyte progenitor goes to monoblast, promonocyte, monocyte, macrophage, over here to myeloblast, promyelocyte, myelocyte, metamyelocyte, and neutrophil. So what it is showing is what kind of like the marker, how dim, how bright, and what kind of markers do these cells have. So as we know, CD34 is a blast marker, and it starts right from the hematopoietic stem cell, goes to the common myeloid progenitor, and also in the myeloblast and the monoblast. I hope you can see my, yeah, you can see my arrow. Then after, the myeloblasts. So the myeloblasts are characterized by having CD117. So you can see there is a CD117. You can see a CD13 can be seen both in the monoblasts as well as the myeloblasts. So you can see CD13 is the same, like plus and plus, both of them in the monoblast and the myeloblast. However, CD33 is dim in the myeloblast and it is there in monoblast. So when we'll show you the normal everything myelomonoblastic, myelomonocytic maturation, you will see all of these things. Coming to HLA-DR. So HLA-DR, you see that it is brighter in the myeloblasts and it is dimmer than the myeloblast in the monoblast. So basically, this again will be helpful when we are going to go through the myelomonocytic maturation. Also remember that the HLA-DR, it kind of vanishes, or no, it is there in promonocytes and monocytes as well, and it is like monoblast, promonocyte, monocyte, all of them have HLA-DR. Coming to monocyte and myelocyte markers, so you can see CD11B, we call it more of a monocyte marker, and you can see as the monocytes mature. So basically you can see over here, the promonocytes have 1+ and the monocytes have 2+. So it is brighter. So as the monocytes mature, and again, you can see myelocyte may have CD11B, but it will not be as bright. So as the monocytes mature, they will gain CD11B. Or the other monocyte markers, they are CD64. And again, you see that CD64 is not there mentioned over here. So CD64 is again another promonocyte, monocyte marker for that matter. And CD14. So CD14, you would see that it is not mentioned anywhere here. So it's not in here. CD14 is a mature monocyte marker. Remember that, CD14, a mature monocyte marker. So this kind of gives us a little bit of overview of what we are expecting when we are looking at the normal myelomonocytic flow. This basically is everything the same. This was actually a PowerPoint that I made for a journal club in my residency. I was like, I should use this one, it's good. Any questions based on this, what we read in this? Anyone wants to discuss? Anyone wants to summarize maybe quickly so that everyone understands what I just blurted out? Rajeshwari, you have something to say about this?
Maybe I can quickly summarize.
Yeah, go ahead.
Right. So one more thing important to remember here is for monocytic lineage, the more specific markers are your 11B, CD14. CD64 can be seen in both myelocytic lineage and in monocytic lineage. And important thing, like Akansha said, promyelocytes do not show HLA-DR, and that helps very handy when you are analyzing your flow for APLs. And other thing is like CD13, when it matures, she's going to show you in the next few slides. When there is maturation in the promyelocytes show CD13, and then they will lose CD13 in the myelocyte stage, and then they again gain it in the metamyelocyte stage and neutrophil stage. but that's pretty much it. And one more interesting, important thing to remember is that CD34 is not a reliable marker for monoblasts. It can be positive, but most of the times in neoplastics, CD34 doesn't help us for analysis.
Right,
Right.
that's very important. And you can see over here that they have mentioned over here as well, they are giving 2+ for all of these, but they are just giving 1+. They should have given +- here.
Yeah, it's almost at the verge of losing it.
Yeah.
Right.
Okay. All right, good. So let's see what was the next that I was going to show you. Yeah, so we can go through quickly the granulocytic monocytic maturation because we are talking about the myeloid series here. So let's see the individual antigens again. This is a wonderful representation by Brentwood, and I wish I would be able to make this in my similars or something in my book, but ultimately we were not able to do that. Maybe we'll make it in our flow book when we do. But what we see here, so normal granulocytic maturation blasts, as we know, they will have CD34 and they will be lost from promyelocyte stage. CD117, however, it kind of dims out in promyelocyte stage, but also still remains. And then there is no 117 following that. As Rajeshri was saying, 13, it kind of dips in the myelocyte stage. So basically, the 13 is kind of brighter in promyelocytes. It dips in the myelocyte stage, and then again takes over, and it's brighter in the neutrophils. CD33 is basically more or less similar, kind of dips a little bit over here, but similar. 66B, most of the labs don't use. I don't know if you guys use it, but I would not go in details of 66B. CD64, so as Rajeshri mentioned, that it can be seen in the myeloid lineages, but usually it is not seen in the mature neutrophils, and it is seen in the mature monocytes. So that's something you should remember. CD15 is more of a mature myeloid marker. So basically it is brighter. So as you go, it is not present in the blast, and it goes brighter and brighter in case of the mature myeloids or mature granulocytes, for example, for that matter, that is neutrophils. So that's CD15. CD65 similarly. CD11B. So as mentioned, CD11B is more or brighter in case of monocytes. However, CD11B can also be expressed in case of myeloid lineage. Then 16. So as you remember, if you have to remember CD15 and CD16. These are the two markers that you have to remember are more of mature myeloid or mature granulocytic markers. Neutrophils, they will express CD15 and 16 very brightly. CD14, very low, if at all present in the myeloid, it will be more in the monocytic lineage. So that's that. Do you have any questions based on this? Anything you want to add?
I just wanted to tell a couple of things. One is the CD66B and the other is CD24. Just remember that those are positive in the neutrophils. As such, we don't use them for myeloid lineage analysis, but when we use it for, when we set up our MRD panels for BALL, they come handy. We'll discuss more about it when we do the BALL panels, but just remember CD66B and 24 are neutrophil markers. One more thing is CD16 is also one of the GPI linked
Right.
thing. So when in PNH cases, it'll be helpful if you don't suspect, but still you see in your myeloid gated myeloid tube that CD16 is lost. It could be because of PNH. So that's something that you can think about.
Good ones. Okay. So this I'm gonna show you, but Rajeshri is also gonna show you because this the way that, so this is from Brentwood, and you know that Brentwood is the guru of flow cytometry He started from Seattle, and he uses Wootlist. So we used at Sloan Kettering, we used to have Wootlist as well. And we were kind of trained into this fashion. However, most of the labs don't have Wootlist, and they have different kind of science, but basically the overall gist remains the same. And Rajeshwari is going to show you some of the signs that are seen in the normal flow. But let me show you let's go through all of this, what we read, to this. Okay. So coming to first thing first, 11B. So 11B, we saw, so basically the maturation is going from here to here. Okay. There is someone that needs to mute. Let me. I'll just mute everyone. Rajeshwari, you unmute when you have to speak. Mute all. Okay. So, yeah. So I was saying that the maturation goes along as the arrow shows, and you can. So you can see the HLA-DR. It kind of dips as it matures, as over here. CD34, as you know, present in the blasts. They are not present in the rest of the markers, so it kind of dips as well. Coming to 11B, so as we know, the 11B kind of is, so we saw over here, so 11B kind of starts in junction of promyelocyte myelocyte, and it kind of is there. So basically you can see over here, it starts from the junction of promyelocyte myelocyte, and it kind of is there in the more mature forms. Coming over here, CD13. So as we were talking about the dipping, right? So the dipping in the myelocyte stage over here. So basically, you can see that CD13 is, so in the more immature forms, it will be there, and then it will rise, as you see over here. So in 13, it will rise, and then there will be a dip. So rise in the promyelocyte and dip in the myelocyte ganglia. So over here, 13 rise in the promyelocyte, dips in the myelocyte, and again it rises up in the more mature neutrophil stages. Coming over here, CD64. So let's see CD64 over, where did it go? Oh, 64 here. So 64 is more, you can see, is present in the promyelocyte stage, and it kind of dips. And then, as I mentioned, 64 is more of a monocyte marker than the myeloid marker, but it can be seen in both of them, as Rajeshwari said. So you can see over here, 64, we were talking that it goes into promyelocyte and then it dips. So let's see. So over here, it is 64 goes into the promyelocyte and then it dips, right? So that makes it clear. I mean, the myeloid maturation is important to understand in order to know the normals versus abnormal. And it's the most difficult. So remember that what you are reading right now, for the beginners, it is most difficult. But if you kind of mug up these things, then you would, and plus you don't need to mug up, like you can just go back and see that kind of cheat sheet. Okay, this is what it looks like. Okay, so this is what it looks like in my case. Like when you're starting, you can start doing that. Okay, let's see. 11B that we were talking about. So 11B, we were saying that it is absent in case of blasts. It is almost absent in the promyelocytes, and it starts getting darker and darker in the brighter and brighter in the mature stages. So we can see over here again, the CD11B, it starts from here and till the promyelocyte stage, and then it starts gaining and to the mature neutrophils. CD64, as we were saying, that it will be present. So let's see 64 over here. 64, I think I already showed. So 64, it will be lost eventually when there is more mature stage. So it will be kind of this curve. CD13, as I showed, there will be a dip. We already talked about that dip in the myelocyte stage. So that's the dip. So this is all saying the same. 16. So 16 is important. 15 and 16, we know. So 15 and 16 are more of the mature granulocytic markers. So you can see over here, the 16 kind of has this like the increasing graph, and it from the promyelocytes, and so you can see over here 16. So it kind of starts from myelocyte, metamyelocyte, and then it kind of brightens in the neutrophils, right? So it starts from myelocyte, metamyelocyte, and it kind of brightens in the neutrophils. And that's that, 16, 14. 14 again. So 14 over here. Where is 14? 14. So again, as I was saying, that it is absent in here, but it is kind of appearing in the neutrophil stage, and that too dim. So if you see, the normal monocytes will be brighter than 14 in the normal neutrophils. So it goes there. However, it is still very dim than the normal monocytes. Did I confuse you even more than you were before?
Oh, that was good.
You have anything to add, Rajshri?
I think like when I show my cases, I'll just
Yeah,
re-emphasize what you said.
Yeah, sounds like a plan.
Yeah, before that, somebody asked in the chat, MPO is important in myelocytic differentiation. Yes, MPO is the lineage defining marker for myelocytic differentiation. But the thing is, MPO is not on the surface, right? It's in the cytoplasm. So most labs do not routinely set up MPO in the regular panels, only if they suspect biphenotypic, or if they don't get the classic myeloid markers. Like if you see some blasts that are positive for 34, 117, 13, and 33, there's no question. But if you see something that is a subset that is positive for 19, or subset that is positive for 3, then they will do MPO. Or if it is negative for everything, then they will do MPO. So usually it is in the add-on tubes. They don't mix the cytoplasmic with this one in the routine panels. All right, so I will start sharing my screen.
Hi, can I ask you a question?
Yeah, sure, Shweta.
Yeah, I was just wondering, can we use 4 and 14 to kind of separate monocytes from the myelocytes? Because these are the markers which are very less prominent in the myeloid and are always in the monocytes. So they kind of gate, like when we gate, can these be used? Because most of the other markers, they have overlapping between myeloid and monocytes.
So for gating purposes, 14 is a very good marker. 4 is not, because when we actually gate, there will be some lymphocytes that you have to eliminate. So you will have to be reverse gating that for that. But otherwise, also there are a couple more things that can help once you the bright intensity of HLA-DR and very bright CD33. These also help for gating purposes.
Mm-hmm.
Yeah.
Thank you.
Okay, so this is just similar to what Akanksha showed in our regular practice. That I just, when we were fellows, our attendings taught us some, shapes that we remember. For 13 versus 16, they used to say it is like in the shape of a Nike swoosh, like the sign you see in the Nike shoes, like the tick mark. So here is how the granulocytes usually mature. The fluorescent green is your neutrophils, or the most mature spectrum of the granulocytic series. And so the end here, this is where the promyelocytes sit. So promyelocytes here, myelocytes here, metamyelocytes here, and then granulocytes here. So it's promyelocytes have 13, they lose 13, and then the myelocytes and metamyelocytes starts gaining 16, and granulocytes have 13 and 16. So that goes in the pattern of a Nike swoosh. Then the other thing is 13 versus 11B. They say it resembles a thyroid gland. You'll see like one limb here, and one lobe here and the other lobe here. So your granulocytes have bright 13 and 11B, whereas we saw before, promyelocytes will have 13, and then myelocytes will lose it, and the metamyelocytes will gain it. So that's how it is here in 13. For 11B, promyelocytes might have a little bit, myelocytes do not have it, and then metamyelocytes and granulocytes will have it. So that looks like a thyroid gland. And then 11B versus 16, 16 is brighter in granulocytes, and 11B and 16 are both brighter in the neutrophils, or the mature granulocytes. So in 11B versus 16, that looks like a water gun or a gun. So you will see 11B increasing as the cells mature, and same way 16 starts, the cells start acquiring 16 somewhere in the myelocyte stage, and then it goes upwards like that. The other thing is like with the blasts. So here the brown population is the blast, and the greens are all the granulocyte cells. So for this is like in a traditional myeloid panel, when you see 13 and 33 versus 34, you can trace the maturation of these cells here. First, you will have these blue population, which are the stem cells that are negative for 34 and 13. And then there is this blast stage where they are positive for 34, negative for 13. And then slowly they will acquire 13 and they'll become positive for 13. And then there is one stage where they're still positive for 34. And then when they become pro-myelocytes, they lose 34 and they have the 13. Same way for 33. So this looks like an inverted C. Same way for 33. There is stem cells that do not have 34 or 33. And then there is blast stage where 34 and 34 is positive, 33 is negative. And then the blast starts to lose 34, like late stage blast, and then your promyelocytes are positive for 33 and no 34. So the same thing you can apply when you analyze for an APL. So APLs are negative for 34. I mean, by definition, negative for 34 and they have 33. That's because they have matured and they have crossed that stage, which is why. Same for 64, like Akanksha said, promyelocytes will have 64 and blasts do not have 64. Same thing. The same way they mature that way. And then as the granulocytes mature, they lose 64. So they come down here. Same thing for HLA-DR. So the blasts will start losing HLA-DR and they will be positive for CD34. And one more, let me see. One more thing is in the stem cells are positive for CD38. Okay, I thought I had 38 here. Looks like I don't. Okay, so here. So the stem cells are positive for CD34, they're negative for CD38, and then slowly they will gain, late stage stem cells just before they become blast, they will be positive for 34. When and in the blast stage, they start getting 38 and they start losing 34, and then slowly they'll become like that. So that's pretty much what I wanted to say. Any questions, anyone? There's one chat. Okay.
Okay.
I'll stop sharing, and Akanksha has more stuff to discuss.
All right. So far, does anyone want to take a pause, think about anything? Have water? Let me have water. Okay. So we directly dived into the harder part of flow. Now, coming to, well, I will not show the promyelocytic leukemia. We'll discuss that when we have the APL. But let's go into monocyte maturation. So monocytes, let's see what they are doing here. CD45 is getting brighter in the monocytes. Okay. CD34, as we saw, it is already dim in the monocytes, and it loses, of course, in the promonocyte and monocyte stage. CD13 again. So remember the 13 dip. CD13 dip was seen in the myelocyte stage here. So here you see it in the myelocyte stage, but CD13 dip is seen in the promonocyte stage, in the normal promonocytes. CD33 is a uniform marker. It will be seen in the blast monocyte and promonocytes. HLA-DR kind of increases in promonocytes. So CD13 dips, HLA-DR increases in the promonocytes. CD64, as I said, CD64, 11B are more seen in the later stages. So basically promonocytes, monocytes, they will have 64 and 11B. As Shweta asked for, it is also there in promonocytes, monocytes, but again, it cannot be used. Another thing is CD16, we were talking that CD16 is a more granulocyte marker. Remember that, more neutrophil, like it's more mature granulocyte marker. So remember that it will be negative in case of blast monocytes and so forth, and it might be dim present in the monocytes. 14, we already said that it is a prominent, like mature monocyte marker, and it is a very important marker. You can use it in your clinical practice. So let's see the monocyte maturation, our hard graph again. When I started my heme path, I used to dread myeloid. I used to really dread myeloid. I used to hate it. So I mean, I would not say that the MDS and everything is still my favorite, but even the MRD and so forth. But at least I have learned a little. Okay, so let's see. The 14. So as I am like emphasizing it again and again so that it goes and stays into your mind, that 14 is mature as the cell monocyte matures, it acquires 14. And CD14 will be the brightest in the monocytes. So remember that. Second is CD64. So 64, as we know that it will also be present in the myelocytes, but it will be also present in the monocytes. So as the cell matures, it gains 64. Coming to 34, as we already know, it will be actually dim to start with in monocytes, but they are saying that the blasts over here can be there, and then it will kind of start dipping, and the promonocytes, monocytes, they will be losing the 34. Coming to HLA-DR, so HLA-DR is more of a reverse U sign. I mean, you don't remember these signs because I would say remember the Rajeshwari signs because those are the ones that will help you in your lab. But this is just for basic understanding. So again, the blasts will be 34 positive and HLA-DR kind of dim, and then it kind of increases. So let's see, we were talking about here that you can see that there is a color difference over here, that it increases in the monocyte stage and then it decreases again. So basically you can see over here the same thing. So it increases in the promonocytes, it decreases again. Coming to 11B. So 11B and 34. So you can see over here the 34 dips and the 11B increases. So that's what I was talking about, that the 14 and the 11B, they are kind of a more mature monocyte markers. However, 11B can also be seen in the granulocytes. So something to remember about that. 14 again, to emphasize again and again, yes, it is more mature monocyte marker. 11B, the same thing over here. 14, 13 is dipping. So you see over here, 13, we saw that it dips in the myelocyte stage in the myelocytes and it dips in the promonocyte stage in the monocytes. So basically it is dipping in the promonocyte stage and it is again coming up. Then 14, this is we saw. 16, so 16, I was saying that it is more of a granulocyte marker, right? So granulocyte marker 15, 16, they are more of the new mature granulocyte markers. So you can see over here, the 16 is uniformly dim in the monocytes. And as this graph is showing that it is gaining 14, but the 16 is uniformly dim. Then coming to the same thing over here, 36, they are showing that it gains 36. I don't know if everyone does 36. I mean, Woodlist has 36, but many of the things I have seen don't have 36 in their labs. So acute monocytic leukemia as well, we'll see later. So far, any questions?
Yeah, I have one more. Which marker do you use for immature monocytes? Like for mature, I get it like 14, 11, we like kind of trending, we see that. But immature monocyte, how do you differentiate it from immature myeloid lineage?
Okay, unfortunately, there is no marker. You can say that 14 will be negative in immature, but that's where it gets very challenging for hemopath people. That's the reason people hate and dread monocytes. So one thing is, like, when you see your CD45 versus site scatter, you see where the population sits in that. If it looks in the more if it looks in the area where the monocytes usually sit, or if they are in the blast gate, that will help. And loss of CD14 helps, but it can be still present sometimes. And sometimes you will see a merge in the population in the blast gate that goes to the monocyte gate. It will be like a continuous population, no separation. That helps. And CD56 helps sometimes, but again, CD56 can also be seen in reactive monocytes, but this would be more homogeneous or more intensity in expression. But you have to use morphology, basically.
Okay. Okay.
Yeah.
Okay, good. All right, so let's see what we can see more. Actually, let's go through this. There is, I mean, we are kind of done with myeloid for now. We'll be going further into myeloid and normal myeloid and how it looks like normal myelomonocytic maturation in a normal cell, in normal plot tomorrow. But I want, because we are doing this, Brentwood, let's quickly do this, the other ones as well. So normal B cell maturation. Before we do that, let's see this. So this is from our book, and this is to show how and where the antigens they lie and what happens to them. So you can see the CD34. So first, let's see what's going on in top here. This is the bone marrow box. This is the interfollicular, that is pre-germinal center box. This is the follicular, that is the germinal center box, and this is post-germinal center box. So basically the pro-B cell, it goes to form pre-B cell, and at that moment of time, it develops a cytoplasmic mu heavy chain. And then that cytoplasmic mu heavy chain, it expresses membrane IgM in case of immature B cells. The immature B cells, they go into the lymph node. So this whole thing is a lymph node. They go into the lymph node. The first cell that happens in the pre-germinal center stage in the lymph node is the naive B cell. So again, these three cells, the immature, pre-B, pro-B, develop a B-ALL, and we have a spectrum of B-ALL that depends on what kind of blast or immature cell it has, and we kind of always talk that it is more mature B-ALL or immature or more immature. Now coming to the pre-GC, the pre-GC, it starts from the naive B cell, and now it has a membrane IgD as well. So membrane IgM that was acquired before, and membrane IgD. Then it goes to extrafollicular B cell blast and a short-lived plasma cell. So long-lived plasma cell will be post-GC, remember, but a short-lived plasma cell can be present in the pre-GC as well. Remember that the mantle cell, it develops from the naive B cell. Now, this pre-GC neoplasm, the mantle cell and naive B cell, they give rise to mantle cell lymphoma. Coming to the follicular phase or germinal center phase. So germinal center, once the extrafollicular B cell blast is there, it goes and first develop a centroblast, and then it goes and develop a centrocyte. So the germinal center neoplasms are more of follicular lymphoma, Burkitt, DLBCL, and Hodgkin's lymphoma. And then the post-GC, the memory B cell and the plasma cell develop. And these are responsible for the plasma cell neoplasm, as well as the memory B cells are responsible for the marginal zones, and the other CLL, SLL is also post-GC, DLBCL. DLBCL can be actually germinal center and activated B cell, though that will come later. But let's see how the antigens, like we are flow people, right? So let's see how the antigens are evolving. CD34 is there in the early, immature or pro-B cell phase. It kind of loses after that. So why is this important? Because in B-ALL, the more mature forms of B-ALL will not have the CD34s. That doesn't mean that they are not B-ALL. Just that's a caveat that you have to remember always. Similarly, the TDT. So there is still an immature B cell that does not have TDT. Remember that. So the TDT will be present in the early immature phases like pro-B cell and pre-B cell, and it will be lost in case of immature B cell. CD10 we always say that CD10 is a germinal center marker, but also remember that CD10 can also be present in the pre-pro B cells. So that's something. LMO2 again, we call them a germinal center marker. We do it for follicular lymphoma. But again, CD10 LMO2 can be present in the early on stages. Newsflash. CD19. So CD19 is more of in the till the germinal center stage, and in the plasma cells, as we all know, the CD19 dips. CD79A, however, is present throughout, right? So CD79A throughout the B cell maturation, even in the plasma cells, it will be present. PAX5, it will not be present in the plasma cells. It is a B cell marker. It will be present in the B cell till the memory B cell, but it will not be present in the plasma cell. CD20, it is not present. It is not a blast or basically immature marker. It is more of a mature marker. So CD20 will start coming in the intermediate or pre-B cell stage, and it will be lasting till memory B cells. It will not be present in plasma cells. So basically we read that PAX5, CD20, not in the plasma cells. CD38. So CD38, we see CD38 in the immature forms over here. And then the CD38 kind of dips in the memory B cell, and again so we call CD38 as plasma cell marker. However, it is also present in the germinal center and a plasma cell. Now BCL6, we know that it is a germinal center marker. HGAL is another like unknown or not very much used germinal center marker. So BCL6, CD10, LMO2, we know that these are the germinal center marker. IRF4 MUM1, it's a nice plasma cell marker that we should remember. IRF4 MUM1 is seen in the plasma cells. CD138 is more seen in the plasma cell, and it is more specific to plasma cell as compared to CD38, which can be seen in the other phases as well. So that's about journey of B lymphocytes. Any questions here?
Hi, I have a question.
Yeah, go ahead.
Does it mean that CD38 can be both in the plasma cells and also in the precursor B cells?
Yes, that's true. That's why when we see the precursor B cell like BLLs, those BLLs, they have CD38. Any other questions? Okay, so let's go ahead with the T cell maturation. Actually, let's go with the Brentwood B cell first. All right, so the Brentwood guru, what he says, the same thing he says. TDT, as we said, it is early. It will not be present in the immature lates. So that's a caveat, always remember. CD34, early, not present in the immature lates. Again, caveat to remember. So basically these three will still be BALL. And there are these two BLLs that will be negative for 34 and TDT. So remember that. CD10. So CD10 dips in the mantle cell, and then basically bright. So we already saw what CD10, the germinal center marker will be more in the follicular center and brighter in the immature. CD38, as we saw that CD38 can come in the immature or late mid stages, and then it is very bright in case of plasma cells. CD19, as we saw that it is not present in the plasma cells, very dim in the plasma cells. It is more in the immature mid stage. CD20, we were talking that it is not present in the early blast. So basically the early BLLs that we have, or maybe like the BLLs that we have, usually they are not CD20 positive. It comes in more of immature late stages and also in the follicular center stages. IgD, IgM, kappa lambda, I mean, those are something that, I mean, that's fine. So let's see over here. So you see over here, the B cell maturation, we have the kappa and the lambda B cells. And I mean, this will more confuse you. Let's see how we can go through this. So do they have, so the TDT again comes in the intracytoplasmic tube, remember that. So we don't have a TDT over here that comes in the intracytoplasmic tube. Now coming to CD34, if they have CD34, they don't have CD34 here. Okay, CD10. So CD10, we saw that it is in the immature early in the follicular center, and CD10 is more in the immature early and in the follicular center. I mean, these, let's not remember this because that will more confuse you. Let's see the ones that Rajeshri will have, because those will be the ones that you'll have in your normal practice. But this is something that you should remember for B cell maturation. I guess we should stop here. I mean, Rajeshri will have something maybe, but T cell maturation and the other things we'll start tomorrow. It's already 4:58. We can give 2 minutes to questions, and we can stop here.
I actually didn't have anything specific for this because I thought when we do BALs, we can do the hematogones, and
Right.
I didn't pull it out for today.
Yeah, you can bring it tomorrow, so we can review it tomorrow. Any questions, anything, any comments you guys have? I mean, we will go through all of this again before we are doing AMLs. We'll go through all of this again before we are doing our Bs and Ts, remember. So there will be a repetition. I have all of this material in the resources already. And when you get a chance, just at your leisure, just go through this. I don't want to dump a lot on you. It's already a myeloid and B cell maturation that has been dumped. Yeah, so do you have any questions, anything to say?
There's a question in the chat box. Do you use CD38 in AML MRD? So I don't think people have a separate panel as such for AML MRD. MRD is a real thing in BLL and TLL, but in myeloid, it's very hard because you normally have some normal myeloid blasts in the marrow. So CD38 is always there in the myeloid tube, but it's very hard to say if it is just for MRD. It's just for myeloid blasts. Yeah, I don't see any other questions.
Thank you very much for excellent presentations.
Very welcome.
Thank you, Dr. Iqbal. Thanks for
Yeah,
joining
thank-
us.
Yeah, sure. Thank you.
Thank you.
Thank you, guys. Thank you for joining.
Thank you very
Thank you so much.
Thank you guys. See you all tomorrow.
Let me
Bye-bye.
know the feedback. Do you think this is good?
Yes, this is very helpful. Very nice presentation. Thank you.
And do we need to be more clear, or is this very complicated for you all?
I like it when you repeat a lot because it's quite new, at least for me.
Yeah.
So that's very
Yeah.
helpful.
Right. Once we are going to go into the cases becomes easy, but these maturation patterns, as I said, I have dreaded always. But once you start seeing it again and again, you start recognizing them.
Yeah, that's the aim, hopefully. Thank you.
Yeah. Thank you.
All right, guys. Bye-bye.
Bye.